Okay. All right. Thank you. Thank you so much, Jill, and thank you to Eterna for allowing me to present my research on structural dynamics of 7S RNA central domain. Some really interesting and exciting stuff coming out of our lab right now. Um, I'll start with RNA structure. Often when people think about RNA, RNA structure chemically, it's often very simple compared to proteins. Proteins have like 20 proteins are typically made up of 20 amino acids, while RNA is made of 4 nucleotides that have a sugar phosphate backbone. So this generally can be regarded as very simple chemically, but in turn, this simple structures, or simple chemical structures, can form really well-structured secondary and tertiary structures. And I believe we've heard a little bit of that from yesterday's presentation. And it gets even more complicated if you have large RNAs where you can have all these motifs like apical loops, Borges, three-way junctions, and so on and so forth. And just to add on to this complexity, some RNAs can dance in a sense that some RNAs can undergo conformational change or conformational dynamics between two secondary structures. And in part, this is because of the fact that RNAs can have the ability to sample different conformations. And in most cases, the lowest free energy structure in most cases is the most biologically relevant conformation, but also you can have other conformations that are a little bit in a higher free energy end that could be relevant. So sometimes in some cases, you can have situations where you have RNAs that are undergoing conformational dynamics or dancing between these two conformations. And just to highlight a few examples of that, I think arrival switches are very good examples of these cases where a rival switch would have one conformation and then it would bind a metabolite and then switch the conformation. And this conformational change upon binding a metabolite and switching has been known to regulate gene expression. So basically, when it binds to a metabolite, gene expression is on, and then when it switches, it turns gene expression off. And a more common example also might be microRNAs. MicroRNAs have been found to be involved in a lot of cancers. And they also exhibit these kind of features where they would have one conformation, but upon binding proteins, they would switch conformations. And to switch gears a little bit, 7SK RNA, which the bulk of my talk would be about. So it's also a non-coding RNA, like microRNAs. 7SK, it's a highly dynamic RNA. It also has been known to exhibit these features where it undergoes conformational exchanges upon binding proteins. So here I'm showing 7SK when it's bound to these proteins, it can downregulate transcription. And then when these proteins are released from the complex, it takes in another conformation where this process upregulates transcription. And I'll go into detail on how 7SK do this in the next slide. But just to appreciate on this slide, you can see that we have almost three different conformations of 7SK where one proteins are bound, and then when proteins are released, and then we have an intermediate conformer. So just to dive into what 7SK does in general, so typically when people think about transcription, we think of it as this linear process where you have DNA, then you make RNA, then you make protein. But actually, there's a lot of regulatory steps within the process. And then one of those steps is in transcription is the arrest of RNA polymerase II. So basically, RNA polymerase II gets recruited to transcription start sites, and then it starts transcription. So when it transcribes about 40 to 60 nucleotides, it undergoes a regulatory step called promoter proximal pausing, where the transcription itself gets halted, partly due to induced by these proteins, DCIF, which are also known as negative elongation factors. So this arrest of polymerase II stores transcription, and for POL2 to be released from this state, it has to be phosphorylated by a protein called PTFP, which stands for positive transcription elongation factor. So PTFB phosphorylates Pol2, and then this DCRF for transcription to continue into productive elongation. So how does 7SK plays into this? So 7SK together with MEPC and LAP7 forms the 7SK SNRMP complex. So basically, this complex regulates the action of PTFB such that they recruit other proteins and together they can inhibit PTFB. So when PTFB is bound to the complex, the 7SK complex, transcription is yeah, so transcription is halted. But when PTFB is released from the complex, then PTFB is able to go ahead and phosphorylate Pol2 for transcription to continue. So uh, the precise mechanism of how this happens, how PTFB is recruited and released, is not very well understood. But it's been known that things like viruses have been known to hijack this resolve this mechanism. Also, sometimes due to self-stress, that can also induce PTFB release. And then one of the things that still remain to be explored is how conformational exchange can lead to PTFB release. So here I'm highlighting these region here. So basically, the stem loop 1 and stem loop 4 of the 7SK have been known to recruit PTFB. So they've been highly characterized and have been well understood on how they play a role in PTFB assembly. But the middle region here, which is the stem loop 2 and the stem loop 3, recently there have been mounting evidence that these regions might be involved in PTFB release. And even in one instance, there was a study that was done where 7SK without stem loop 3 can recruit PTFB or can bind PTFB, but then it wasn't able to release it. So all these were indications of the importance of understanding how stem loop 3 its involvement in PTFB release. Additionally, 7SK has been known to regulate other processes in cells. So it doesn't just regulate transcription elongation, but it also regulates enhanced RNA initiations, also transcription elongation of small nuclear and nucleolar RNAs, and also has been found to regulate axonal growth and differentiation. So it does a lot of things in cells. And then the misregulation of this process of PTFB release and recruiting has been linked to a lot of diseases, mainly cancer, developmental disorders, heart diseases, and so on. So there's a growing need to understand how this mechanism really works and what role does stem loop 3 for us. In the case, what role does stem loop 3 does in this process? So our main question was, what is the structure and dynamics of stem loop 3? Like I mentioned, there's been a lot of studies on stem loop 1 and stem loop 4, which are the PTFB assembly areas. But then when it comes to the PTFB release areas, there hasn't been a lot of studies done. So our goal was to determine the structure and dynamics of 7SK and stem loop 3. And then to do this, we use two different methods, mainly two different methods, which is nuclear magnetic resonance spectroscopy. So Talbot alluded to this a little bit, but NMR is really a great tool to probe RNA structures. And orthogonally, we can use, we also employ DMS chemical mapping in order to understand the secondary structure of RNA. So with NMR is really a great tool. You can really probe a wide variety of atoms. So here I'm highlighting just the areas. Yeah, you can do a lot with NMR, but I'm highlighting just the ones that were the ones that I have with the results that I'm going to show. Mainly, you can with NMR you can probe base pairing interactions and base stacking interactions using these MNO protons. Also, you can find out how many adenines you have, assign adenines, and so on and so forth. With DMS, however, you can which it's kind of complementary to NMR, where you can probe RNAs based on solvent accessibility. So if a particular nucleotide is exposed to the solvent, mainly A's and C's, so DMS adds a metal to the N1 of A and then to the N3 of C's. So if these nucleotides are solvent exposed, DMS can be able to modify it, but if they're in a base pair, that accessibility is sequestered and then DMS will not be able to modify it. So with this DMS can make a mapping coupled with sequencing and structure prediction, we can be able to determine our RNA structure. And then we use a DMS technique called DMS MAPSIC, which was developed by Sylvia Ruskin. It's a very robust technique that has been found to have great results. So to start with our experiment, so like I said, our goal in the first place was to determine the structure and dynamics of 7-fs stem loop 3. So we decided to look at this region of stem loop 3, which is here showing the secondary structure. The reason why we went for this region was this region has been known to bind many proteins and also has sites for post-transcriptional modifications. So we decided to look at this region, and we started with an NMR basically to probe for the adenines, the C2 of adenines. And what we found was that basically with this technique, each peak should be 1A, and then so the expectation is that the number of peaks you have here should match the number of A's you have in your secondary structure. And expected A's or peaks that we were expecting were 16, but then we observed greater than 21 peaks. So which was concerning because that points to the fact that there is either conformational exchange or RNA is degrading. And we've done several tests to make sure that we've run native and denaturing gels to check our integrity of our RNA, and we confirm that our RNA was indeed not degrading. So what we did was we moved to to probe other regions of our RNA. So basically, we decided to probe for base pair interactions to see if indeed we see any conformational exchange. So what we observed was that we did a temperature titration from 5 degrees to 35 degrees. And what we observed was that, so basically we're probing for this MNO hydrogens in our structure. So basically, each base pair should be a peak. Each base pair should be a peak. So what we observed was that as we increased temperature, we saw some peaks becoming sharp, while some peaks become. So ideally temperature melts base pairs. So if you increase in temperature, you would expect that your base pair should be melting and not forming. And what we observe is the opposite, that as we increase temperature, our base pair is forming instead of melting. So that itself, it's a common phenomenon in NMR called chemical exchange, where basically at lower temperatures you have your RNA, it's in a slow exchange, and as you go up in temperature, you're pushing it to a more fast exchange, so the peak become a lot sharper. So this was one of the early first indications of conformational exchange in stem loop 3. So we decided to turn to DMS MAPSIC since it was supposedly tested on RNAs, and then it can also DMS MAPSIC coupled with DREAM, again, which was developed by the Ruskin lab. You can determine alternate conformations of RNA. So we use this technique to probe for our stem loop 3 construct. And what we found was that the DMS MAPSIC was also consistent with our NMR data. As in, so here, again, DMS, we are modifying just A's and C's. So that's why for the G's and U's, we're not seeing any signal. So basically, from the DMS MAPSIC, we found that this C, which is in a base pair, exhibited a moderate to a high reactivity. And also these A's here, which are in a base pair, also, have a moderate reactivity. And then this A here, which is also in a base pair, also showed a moderate activity. So this was an indication that the DMS MAPSIC data wasn't just consistent with one secondary structure, but instead it's also showing conformational exchange. So, like I said, the Ruskin lab developed this pipeline where you have DMS mapseq and then Dream. You do a Dream clustering. Basically, it's a pattern matching algorithm that looks at pattern within the DMS signal and then clusters the RNAs between different populations. So we decided to employ this technique, and lo and behold, what we found was that stem loop 3 was clustered into two clusters. So we named them SL3A and SL3E. Sorry, the cluster 2 should be SL3A. That's a mistake. So yeah, we named them cluster 1 is SL3E and then cluster 2 is SL3A. So basically, the cluster 1 matches the secondary structure that has been seen widely in literature, especially most recently. And then cluster 2 also has been found in literature, but then I will show that later. But then basically what we found was that we have two populations, basically E and A. And then both of these populations, basically um, the difference is that this C221 that I highlighted before, here is in a base pair in the E state, which I've shown before. But then in this cluster 2 is in an A state. It's part of a loop, so it exhibited a high reactivity in cluster 2. Additionally, A251, which in the A state is also in a base pair, but which in the E state is in a base pair, sorry. But in the A state, it's part of this bulge here, also exhibited a really high reactivity. And then additionally, these three A's, so basically 220, 229, 30, and 31 here, here they are part of a loop, but in here they sequestered in a base pair. So you can see the difference there additionally as well, where when they're part of the loop, they have higher activities compared to when they're in a base pair. So this coupled with the NMR data, was an indication that stem loop 3 of the 7 escapes undergoing some conformational exchange between these two states. And then we decided to explore this in the full length 7SK. Previously, we've been using the distal construct, a small construct of the 7SK. But we decided to explore this in the full length 7SK. And what we found was that, so this is the average reactivity before dream clustering. But when we did the dream clustering, we also observed that we have these two populations, roughly equal populations of these two conformers. So from this data, we develop a model where we developed a model where 7SK stem loop 3 is undergoing conformational exchange. So basically, it's undergoing almost an 8 base pair rearrangement between these two conformers, so A and E. And what's even more interesting is that these two secondary structures have previously been reported in literature from different independent studies. So here I'm highlighting some of the papers that have reported this before. One is from an evolutionary study, and then a more recent one, Talbot is here, it was from the Talbot lab where they reported this secondary structure. And also in 2022, the Weeks lab reported this type of secondary structure. But additionally, this other conformer that we are also seeing has been reported in literature from the early days of 7SK studies, like from 84, 91, and so on and so forth. So this is pretty interesting because, like I said, I mean, both of these secondary structures have been reported in literature, but they have not been found to interchange or undergo conformational exchange. So we decided to explore this a little bit more. And what we did was just trying to understand this conformational exchange. So we decided to see if we can log these states. So basically, if can we stabilize one state over the other? So to do that, we decided to start with the top portion of stem loop 3. Basically, we just changed the sequence boundaries. Here we're going from 22 to 222 to 255. And we observe that when we change this, just by changing the sequence boundaries, we can stabilize the A state. Additionally, if we change the sequence boundary. So if we go from 221 to just 253, we observe that we can stabilize the E state. And I have an NMR data for this here, and this clearly depicts this. So when we overlaid the two states, the two structures. When you overlay the two on the distal construct, we observe that it perfectly overlays with the stem loop 3 distal. Stem loop 3 distal being the stem loop 3 cell, and then the green is the A, and then the purple is the E. So additionally, we found that we hypothesized that, okay, can we do mutations? Instead of changing the boundaries, can we do mutations to stabilize one conformer over the other? And then we hypothesized that if we change this C224, which has a CU bulge there, if we change it to an A, can we stabilize the E state? Additionally, we also hypothesized that, okay, if we delete the C221, would that stabilize the A state? And lo and behold, when we here we're probing for the A2, like I mentioned before. Here we're probing for the A2, so we expected to see just peaks for the A2. And what we observe is that when we overlay these mutants on the distal construct, we observe that it perfectly these mutants perfectly stabilize the E and the A state. And with few differences in chemical environment, which can be like, for instance, for this A251, which can be attributed to experiencing a different chemical environment, because this this A251 here, basically, it's going from a CU bulge into a AU base pair. So that stabilization there is giving it a different chemical environment. So that's why we see a shift there for a particular residue. But everything else clearly overlays really well. So we decided to see if we can fit these two states. So basically, using these mutants, can we use them as fingerprint? And this was really inspired by the work of Hashimi Lab. So basically, they have this technique which they employ with NMR called mutation chemical shift fingerprinting. So basically, they would use mutations to fingerprint excited state conformations that they typically observe, or alternative conformations that they typically observe. So we decided to see if we can use the same technique, but instead of with NMR, use it with DMS. So based on these mutants that we made, the ELOC mutant and the ALOC mutant, we did chemical mapping studies to see if these mutants clearly can be used as fingerprints for the SL3, for the E and the A states. And then when we did the chemical mapping studies, here I'm not showing the chemical, but I'm just showing the correlation between the two states. So basically, the mutant ELOC and the E cluster that we observed from the distal had a really nice correlation. So, and then additionally for the ALOC as well, when we did a DMS map seek and then compared to the SL to the A cluster, we also observe a really nice correlation. So we decided to use these two mutants as fingerprints to see if we can fit the distal construct to a two-state model. So to do that, we decided to couple the mutational DMS fingerprinting with linear combination. Basically, we benchmarked this on the distal construct, where we took the mutational fraction from the distal construct, and then use the mutational fraction of the mutants, that is the ELOC and then ALOC, and then did a linear combination, and then fit that to the distal construct. So basically, to see if we can take these two fingerprints to fit it to a two-state model. So here, as you can see, this is the result that we got from the linear combination. And what we observe is that the populations that we get closely matched to the populations that we had from the dream clustering, which was great. Additionally, the linear combination of the two mutants nicely fit to the distal construct. So on top of just doing it on the distal construct, we also did the same on the full length construct, full length 7SK, but just on the stem loop 3 region. And additionally, just like with the distal construct, this also nicely fits the data that we have, and it's pretty consistent with the results that we have from the dream clustering. So from all of this, we from all of this, we came up with this model where, like I mentioned, stem loop 3 is undergoing conformational exchange between these two states. But what's even more interesting is that these two states are roughly equal populations. So these type of structures is what you term as bistable secondary structures. Basically, you have two stable secondary structures that are interchanging in a slow exchange time scale. And then what's even more interesting is that these type of like bistable structures have been found to be very common in riboswitches, and then but that typically involve switching between a single strand and a hairpin. So this is actually one of the few rare cases where you would have a bistable element part of the stem. So that's pretty interesting, and it's something that we're still exploring to understand how, or like what's the mechanism, because as you can see, it involves the 8 base pair rearrangement, which is the huge energetics to overcome in order to achieve this, to go between these two second conformations. So overall, our hope is that these two models would make us really understand how stem loop 3 is involved in PTFB release. Like I said in the beginning, these regions of stem loop 3 have been involved in protein binding, and this U250 has been involved in post-transcriptional modification. So it's highly possible that this conformational exchange has some role to play in that process. So that's an active area of research in our lab where we're exploring how proteins bind to these two conformers, and then so on and so forth. With that, I would like to thank my supervisor, Dr. Icon, for the support, and then our collaborators. We've been collaborating with the Yeselman lab to do some of the DMS maps 6 stuff. And then my lab members, and then just to acknowledge the funding, in the early days, this project was funded by the Nebraska Center for Integration Structural Biology. And then now it's being funded by NSF and NIH. That's all I have. Thank you.